What if I told you that right now, as you're reading this, there's a technology so powerful it could rewrite the very fabric of life itself? A technology that makes genetic engineering look like child's play, that could eliminate diseases before they even exist, but also create possibilities so terrifying that some of the world's leading scientists are calling for it to be banned entirely. This isn't science fiction. This is happening right now in laboratories around the world, and the question isn't whether this technology will change everything we know about biology, it's whether we should let it. Picture this. Imagine you could edit a living organism's DNA with the same precision you use to edit a document on your computer. Cut, copy, paste, delete. But instead of words, you're rearranging the very code of life. Now imagine that every edit you make doesn't just affect one organism, but potentially every single member of that species, forever. Every mosquito, every mouse, every plant, every microbe. One edit, billions of consequences. Welcome to the world of gene drives. To understand why gene drives are simultaneously the most promising and most terrifying biological technology ever created, we need to take a step back and look at how nature normally works. In traditional genetics, when two organisms reproduce, each parent contributes roughly half of their genes to their offspring. It's like flipping a coin, heads or tails, mom's version or dad's version of each gene. This is Mendel's law, the foundation of inheritance that's been true for every living thing on Earth for billions of years. But what if you could load the dice? What if you could ensure that a particular gene gets passed on to 100% of offspring, not just 50%? That's exactly what a gene drive does. It cheats the fundamental rules of inheritance. Here's where it gets mind-bending. Scientists have discovered a way to build genetic systems that essentially copy themselves into the DNA of every offspring. When an organism with a gene drive reproduces, instead of the normal 50-50 split, the gene drive actively seeks out and replaces the natural version of the gene in the other parent's DNA. It's like having a genetic copy machine that ensures its own survival and spread. The implications are staggering. Release just a few hundred modified mosquitoes carrying a gene drive into the wild, and within a matter of generations, you could potentially modify every single mosquito in a region. Every single one. Now, before your mind jumps to the horrifying possibilities, and trust me, we'll get there, let's consider the incredible potential for good. Malaria kills over 600,000 people every year, most of them children under 5. What if we could use gene drives to make mosquitoes unable to carry the malaria parasite? We could potentially save hundreds of thousands of lives annually. Or consider invasive species that are destroying entire ecosystems. What if we could use gene drives to reduce their populations or make them less harmful? What about crop pests that cause billions of dollars in damage and threaten food security for millions of people? The science behind this is elegantly simple yet mind-numbingly complex. Researchers use a molecular system called CRISPR-Cas9. You might have heard of CRISPR as the gene editing tool that won the Nobel Prize, but in gene drives, CRISPR becomes something far more powerful. The system includes the molecular scissors that cut DNA, the guide that tells it where to cut, and most crucially, a template for what should be inserted in place of the cut section. When reproduction happens, this entire toolkit gets activated. The gene drive seeks out the corresponding location on the partner's chromosome and replaces it with its own version. It's like having a biological photocopier that not only makes copies of itself, but actively seeks out and overwrites the original documents. But here's where the story takes a darker turn, and where some of the world's most brilliant scientists start losing sleep at night. Gene drives don't respect borders. They don't ask permission. They don't care about your political system, your environmental regulations, or your ethical concerns. Once released into the wild, they could spread globally, potentially forever. There is currently no known way to recall a gene drive once it's been released into a wild population. If you enjoy fascinating stories like this, don't forget to like this video, subscribe to our channel, and turn on the notification bell so you never miss our content. And tell us in the comments where you're watching from. We'd love to know. Think about it this way. Imagine if one country decided to release gene-drive mosquitoes to combat malaria, but those mosquitoes carried an unintended mutation that made them more likely to carry a different disease, or that somehow disrupted the ecosystem in an unexpected way. Those consequences wouldn't stay within that country's borders. Mosquitoes don't carry passports. This has led to what researchers call the gene drive dilemma, the same mechanism that makes gene drives so potentially powerful for solving global problems also makes them potentially catastrophic if something goes wrong. And in biology, things go wrong more often than we'd like to admit. Consider what happened with the cane toad in Australia. In 1935, about 100 cane toads were introduced to control crop pests. Today, there are over 200 million of them, and they've become one of Australia's worst environmental disasters, poisoning native animals and disrupting ecosystems across the continent. Now imagine if those toads had been equipped with a gene drive that ensured their rapid spread and reproduction. The ecological catastrophe would have been immeasurably worse, but the concerns go even deeper than ecological risks. Gene drives represent a form of biological power that humanity has never possessed before. For the first time in history, a small group of people could potentially make irreversible changes to entire species without the consent of anyone else on the planet. What if a gene drive designed to reduce mosquito populations worked too well, collapsing entire food webs that depend on mosquitoes? What if gene drives became weapons used to target crops that feed entire populations? What if they were used to create biological weapons that could spread through wild animal populations? The more scientists study gene drives, the more they realize we're dealing with something that could reshape the entire planet's biosphere. Some researchers have proposed that gene drives should be subject to the same international oversight as nuclear weapons. Others argue that the humanitarian benefits are so enormous that the risks are worth taking. Here's the paradox that keeps scientists awake at night. The same properties that make gene drives potentially world-saving also make them potentially world-ending. Their power lies in their ability to spread and persist, but those same qualities make them nearly impossible to control once released. Recent research has revealed just how complex and unpredictable gene drives can be. In laboratory studies, scientists have found that gene drives can mutate, evolve, and behave in unexpected ways. Some drives that were designed to spread rapidly through populations instead died out after just a few generations. Others, that were designed to be temporary, became permanent. Evolution, it turns out, doesn't always follow our carefully laid plans. But perhaps most unsettling of all, researchers have discovered that gene drives can potentially evolve to become more aggressive and harder to stop. Natural selection favors gene drives that spread more effectively, which means that over time, any gene drives released into the wild might become better and better at overriding natural genetic systems. This creates what scientists call an evolutionary arms race. As we develop better containment methods, gene drives may evolve better ways to escape containment. It's a biological game of chess where the stakes are entire species and ecosystems. The current state of gene drive research is a careful balance between promise and caution. Scientists are developing what they call safeguards, genetic kill switches, drives that burn themselves out after a certain number of generations, and containment systems designed to prevent accidental release. But each of these solutions brings its own set of challenges and uncertainties. Some researchers are working on reversible gene drives, systems that could theoretically be undone by releasing a second gene drive designed to counteract the first. But this approach assumes that the reversing drive would spread as effectively as the original, which may not always be the case. Others are developing local gene drives that are designed to spread through a population, but then disappear over time. These could potentially provide the benefits of gene drives while minimizing long-term risks. But the technology is still in its infancy, and the behavior of these systems in real-world environments remains largely unknown. The international scientific community is grappling with unprecedented questions about governance and consent. Who has the right to make decisions about releasing gene drives that could affect the entire planet? How do we weigh the potential to save hundreds of thousands of lives from malaria against the risk of unintended ecological consequences? How do we ensure that this technology is used responsibly when its effects could be irreversible? Some countries are moving forward with gene drive research and field trials, while others are calling for moratoria or outright bans. The lack of international consensus means that gene drives could potentially be deployed by some nations, while others object. A scenario that could lead to serious diplomatic and environmental conflicts. What makes this particularly challenging is that unlike other powerful technologies, gene drives could potentially be developed with relatively modest resources. The core technologies, CRISPR and basic molecular biology techniques, are becoming increasingly accessible to laboratories around the world. This democratization of genetic engineering means that gene drives can't be controlled in the same way as nuclear weapons or other traditionally restricted technologies. As we stand at this crossroads, we're forced to confront fundamental questions about our relationship with nature and our responsibility as a species. Gene drives represent perhaps the most powerful biological tool humans have ever developed, but they also represent a level of planetary responsibility that we may not be ready for. The debate isn't really about whether gene drives should exist. They already do, in research laboratories around the world. The question is whether we're wise enough, careful enough, and collaborative enough as a global community to use them responsibly. What's clear is that gene drives will force us to think about biology, ecology, and global cooperation in entirely new ways. They challenge our assumptions about the boundaries between natural and artificial, about national sovereignty and global responsibility, about the pace of scientific progress, and the time needed for careful deliberation. The future of gene drives remains unwritten, but one thing is certain: the decisions we make about this technology in the next few years will echo through generations, possibly forever. We're not just editing genes, we're editing the future of life on Earth. And if you enjoyed this story, you will definitely love the next one appearing on your screen. Don't forget to subscribe, leave a like, and activate notifications. See you in the next video.